What Mendel called hereditary factors are located on chromosomes. A century ago, this relationship was not so obvious, and most people were skeptical until evidence mounted that there was a physical basis for Mendel's predictions, or these laws that he had established in the behavior of chromosomes. So it wasn't until around 1902 that we began to see parallels here. Chromosomes and genes are both present in pairs. Homologous chromosomes separate and alleles segregate. Fertilization restores that paired condition. So we started seeing all of those inheritance, segregation, independent assortments, all of those things became reflected in the behavior of the chromosome. Basically, it became the chromosomal theory of inheritance, where genes occupy specific locations on chromosomes, and then segregation and independent assortment apply during meiosis. The behavior of homologous chromosomes account for the segregation, and the behavior of the non-homologous chromosomes can account for independent assortment. Be definitively linked to chromosomes, however, were sex-linked traits. This comes from the work of Thomas Hunt Morgan and his research assistants. When they crossed white-eyed males with red-eyed females, all of the F1 offspring had red eyes, suggesting that the red eye allele was dominant. However, the white eye allele appeared in only the F2 males, not the classic 3-to-1 relationship that was expected. All of the F2 females and half of the F2 males had red eyes. So Morgan reasoned that the gene with the white eye mutation was on the X chromosome, and there was no corresponding allele present on the Y. This relates to humans because fruit flies, like humans, have that XXXY chromosomal relationship. So it wouldn't have worked necessarily with other organisms that might have different sex inheritance. But the pair of sex chromosomes in females consists of two X chromosomes, each carrying the same genes. The pair of sex chromosomes in males consists of one X chromosome and one Y chromosome. So during meiosis 1, when chromosomes separate, each gamete from the female parent is receiving an X, but the gametes from the male can either receive an X or a Y. Males are hemizygous for the X. They only have one, and they're supposed to, not homozygous or heterozygous, hemizygous. The sex-linked traits led to a great deal of new information, and we know now that there are many traits that are X-linked, and there are also traits that are Y-linked. The human X chromosome contains approximately 1,100 X-linked genes that we're studying for a variety of factors. The Y chromosome carries significantly less. But in females, this isn't really much of an issue, this whole extra set of DNA, because during female development, one X chromosome per cell condenses into a compact form called a bar body. It's an inactive form of the X chromosome. So it's called X inactivation or lionization for the female researcher who discovered this. This condensed bar body chromosome is reactivated in ovarian cells that produce eggs, and that continues on. So Mary Lyon, who figured this out, demonstrated that selection of the X chromosome occurs randomly, and as a consequence, females consist of a mosaic of two types, some with an active paternal X and some with an active maternal X. So that's where like the fur color of a tortoiseshell cat comes in. That's only going to be in a female cat because they have two X chromosomes, or with certain mutations where they have extra X chromosomes. The eukaryotic cells' genes are located on nuclear chromosomes. We have to remember that there are extranuclear genes, genes that can be found outside of the nucleus. So for us, that's the mitochondria, but for plants, for example, that's both the mitochondria and the chloroplast. Because these genes are only coming from the egg cell, the female cells, these genes can only be inherited matrilineally. So in most plants, a zygote receives all of its plastids from the egg cytoplasm, and so the maternal parent determines coloration, as well as a wide variety of other factors. Several rare human disorders are produced by mutations to mitochondrial DNA, primarily affecting ATP supply, nervous system and muscle energy formation, neuropathy components. So it's a whole variety of issues. New mutations can gradually accumulate, and some researchers think that these mutations play a role in the normal cell aging process. So if you look at Punnett squares of matrilineal inheritance, you can see that it only passes on when the female has the trait, not when the male passes. This is not a guarantee. Just because these cells are selected doesn't mean you're going to necessarily get mutated mitochondria or chloroplasts, so it's not going to be 100% passage. So you can see some examples on the bottom where not all of the offspring received the trait. But if the trait is going to continue to the next generation, it must pass through a female parent. Otherwise, it's not an extranuclear trait. Your direct DNA, there's also a variety of other factors that can alter inheritance patterns, and we refer to these as epigenetics. These are heritable changes in gene expression caused by mechanisms other than changes in DNA. One example of this is genomic imprinting, which basically the parent a gene comes from matters. Epigenetics affects how genes are read by cells and subsequently whether the cells should produce relevant proteins, but it has nothing to do with the DNA sequence. Another way that you can have an epigenetic impact is by looking at things like DNA methylation and DNA acetylation. Basically, they're altering the charges of the histone proteins, causing it to be more dense, more compact, or less dense. Or with DNA methylation, you're directly tagging with a methyl group so that the gene is unable to be accessed by an enzyme. So epigenetics are going to be phenotypic effects that depend on whether they are inherited from the mother or the father, like with imprinting, or alteration in the patterns of inheritance because of alterations in condension. It's incredibly complicated, but we understand now that this is something to do with a wide variety of traits that don't reflect mutationary changes, so cancer, autoimmune disease, mental disorders. A great deal of work is going into what this really means in the future. The assortment of chromosomes produces genetic recombination of unlinked genes. So if genes are unlinked, they're going to be inherited separately. If genes are linked or on the same chromosome, they're going to be inherited together. So we can use chi-square, we can use these predictions to determine whether this trait is something that is necessarily something that is linked or not. Okay, so if we look at the example on the right, you would predict a 1-to-1 to 1-to-1 relationship if the genes are not linked. If the genes were linked, you would expect a 1-to-1 to 0-to-0. Looking at the results, we have 965 to 944, then to 206, and then to 185. That looks like a 1-to-1 relationship, but looks like is not enough. You're gonna need to do stats, and that's where chi-square would come in. Here is that we have these two different traits. We see that we have this B trait and we have this VG trait. The B trait is for black skin or black coat of the fly, whatever you want to call that, and the VG is for the vestigial wings. If you look on the right, you can see our inheritance. If they are linked, like we think they are, you would expect 100% of them to look like the parents, but they don't. There are some that are different. That means that there is some level of recombination. There is offspring that have a new combination of traits. What's making this happen? Well, it's crossing over during meiosis. It could be meiosis 1, less likely to be meiosis 2, but still technically possible. And there are a set of proteins that orchestrate this exchange from one maternal and one paternal, and that recombination accounts for those differences that we accumulate. The subsequent events of meiosis will then distribute those throughout the gamete to produce that recombinant. But now that we understand why they're not matching that exactly, we can then move forward to analyze it with math. Complete the chi-square, which we're not doing right now in the video, you will find that statistically they are linked traits. But there are those ones that are different. We had 206 that were gray vestigial, 185 that were black normal. Those are not the same as the original parent. So the chi-square told me that that difference is significant. But what does that difference really mean as far as the genetics? Well, we can use this so-called recombination frequency, or the amount of recombination, to construct a genomic map. So recombination frequency is a very simple formula that's not provided to you on your math reference sheet. You take the number of recombinants, aka the ones that are different from the parents, and divide those by the total number of offspring. Multiply that by 100 and you'll get the recombination frequency. This recombination frequency is a percentage of the genome and can be used to construct map units. So between these two traits, there is 17% of the genome. So between B and VG, there is 17%. If you keep doing this for a variety of genes, you'll get a series of percentages, recombination frequencies, and you can construct a map of the genes. So you look at the example on the right, we've got three different genes, A, B, and C. If we're looking at the amount, that line is represented there so you can draw your own map of what you expect. Pause here for a second, see what combination you can find. Start with the largest one and work backwards. Okay, so if 18.5 is that relationship, we can see that 13.2 and 6.4 will add that into that broader focus. So A and B, B to C, that would make sense. Okay, now that actually adds up to 19.6. Why does that not add up perfect? Real data doesn't work out perfect. Unfortunately, there are double crossing over events and other sources of errors that can make it not add up perfect. So you're looking for the closest match you can find. Most of the sample problems will be made up data, so the numbers will work out a little nicer, but they don't necessarily have to. Outside of crossing over creating differences, you also have mutations. Physical and chemical disturbances, as well as errors during meiosis, can damage chromosomes in major ways or alter their numbers in cells. Major chromosomal damage in humans and other mammals often lead to miscarriage, and individuals exhibit various developmental disorders. Plants tolerate this a little bit better than animals, so you're going to see more plants that are having wrong numbers of chromosomes. But if chromosomes don't separate properly, you end up with non-disjunction mutations, which means you're going to have the wrong number of chromosomes or an aneuploidy mutation. The most well-known example of an aneuploidy mutation would be trisomy 21, which is Down syndrome. Trisomic cells have 3 copies of a particular chromosome, and that is what's going on there. Okay, you can also have things like trisomy 18, which is Edwards syndrome, and trisomy 13, which is Patau syndrome. Overall, these are referred to as autosomal or somatic aneuploidies because they're looking at the body chromosomes. If you start looking at the X and the Y chromosome, that's going to be the allosomal or gametic aneuploidies. That's going to be things like Turner syndrome, where you only have one X, or trisomy X, where you have 3 of them. Klinefelter's is an XXY. Jacob syndrome is an XYY. There's a variety of them, but it's an autosomal aneuploidy if you have the wrong number of a somatic chromosome, and it's an allosomal aneuploidy if you have the wrong number of a gametic chromosome. More than 2 complete sets of chromosomes are polyploid. So polyploidy is going to mean that you have more sets. Plants can survive this, animals can't. And we find that there are a variety of organisms that are polyploid on purpose. We have purposely inbred them to produce these structures, like seedless watermelons and bananas. They are triploid. Okay, if you have something like strawberry, for example, it's octoploid. Overall, you have a 2N zygote. If it fails to divide after replicating, a tetraploid embryo results, and that's going to get that broader focus. Triploid watermelon seeds, for example, are sterile because you need pairs of chromosomes to form seeds. That's why animals and plants always have multiples of 2. So if you need to produce a seedless plant, you need it to be in an odd number relationship. So we see that that's something that we take advantage of as a tool in modern plant breeding. To be whole chromosomes, though, you could have chromosomal alterations that are parts or subsets. Deletions are pretty self-explanatory. A chromosomal segment is removed. Duplication, a chromosomal segment is copied. Inversion, a chromosomal segment is inverted, is flipped around. So if you look at the picture, that makes a little bit more sense. The chromosomal fragment reattaches in a reverse orientation. The more odd one is a translocation. A translocation is a non-homologous crossing over event. So crossing over is between homologous chromosomes and has minimal impact, but translocation is between non-homologous chromosomes. So one example overall here, as like Cri du Chat, for example, is a specific deletion chromosomal 5, And individuals with that disease have mental disorders, smaller heads, unusual facial features, and have a cry like that of a distressed cat, which is where the name came from, and it's fatal in early infancy. There are several human disorders due to alterations of chromosomes that are more common to be something in an adult. That would be something like, say, Huntington's, for example, which is a duplication that alters an enzyme that causes the death of nerve cells. Philadelphia syndrome is a translocation that causes a type of leukemia. Okay, it's between chromosome 22 and chromosome 9, that Philadelphia chromosome relationship. There are a variety of types of schizophrenia associated with this. So chromosomal alterations can get incredibly complicated. It doesn't have to be whole chromosomes. It just needs to be essential genes. A lot of these are lethal, but some of them survive with a characteristic set of symptoms, and we call those diseases syndromes. The only real way to test for these syndromes is prenatal diagnosis and genetic testing. Many hospitals have genetic counselors to provide information to prospective parents who are concerned about a family history of a specific disease. And there's really two ways you can do this. You have amniocentesis. Amniocentesis, a sample of the amniotic fluid, that fluid around the baby, is removed and sent to a laboratory that is then going to be tested through karyotypes. Um, Chorionic villus sampling is similar, but you take a sample of the chorion. Okay, so the chorion is going to allow that faster karyotyping because it can be formed earlier in development, but it's going to be removing a piece of fetal tissue. Recent techniques allow us to do this a little bit safer, but there is still going to be a risk no matter what. So amniocentesis or CVS could be considered in situations in which the parents are at an increased risk of having an infant with a genetic disorder, and they may be useful, especially if you're over the age of 35. But it's helpful, but there is always going to be risk because it's going into medical testing, surgery, a variety of issues. So it's something to definitely discuss with your doctor.